Hello everyone, welcome back for more biotechnology. Let's finish up chapter 2 here with a very short video just to kind of put all the information, all the foundational stuff that we've talked about in this chapter into some perspective. So we've talked a lot about the structure of both proteins, nucleic acids, and how those kind of fit into the central dogma that we've talked about and that we're going to continue to talk about throughout this semester. So historically with biotech, the discovery of being able to manipulate DNA and produce proteins to create this recombinant DNA technology, really a major breakthrough, is really a game changer in that regard. So what this allows us to do is take DNA sequence from various organisms and combine them together in various ways to produce protein products that ordinarily would not exist. So if you kind of think of a gene and its protein product as natural, recombinant DNA and recombinant proteins might be considered unnatural, just without the negative connotation that's associated. The nice thing about this, and what you're seeing here, is kind of another thing that we've seen before, the flow chart of how you might go about making recombinant insulin. Now, obviously the case here is that E. coli cells do not make insulin, but because we introduce the insulin gene, we can manipulate them into doing so. And since they don't naturally make insulin, we would call that recombinant insulin. They're making a protein that they do not normally make because we gave them a DNA sequence that they don't normally have. And we've already said that it's the universal nature of DNA, the fact that all cells speak the DNA and genetic code language, that allows this technology to work. So let's look at a specific example. I think you're going to think this is really cool, or at least I hope you do. So let's talk about a protein called green fluorescent protein, or GFP. This is a bioluminescent protein that is made by certain marine animals, like the jellyfish A. victoria. So you're kind of seeing this jellyfish here, and it kind of seems to glow. So that is because of the presence and production of this GFP protein. Now let's talk about a protein that is normally found in the matrix of mitochondria. This is called heat shock protein 60, or HSP60. Has nothing to do with GFP, just totally an aside here. But let's think about these two proteins together. What could we do with these two proteins? Well, what if we wanted to make it so that the mitochondria inside of a cell fluoresce green? Mitochondria are not usually glow in the dark, but through recombinant DNA technology, we can make the mitochondria glow in the dark like this. So if we can isolate the genes for both of these proteins, the gene for GFP and the gene for HSP60, and splice them together, we can make a version of heat shock protein 60, which we know is in the mitochondria, and it is going to be attached to the green fluorescent protein, which is going to make it fluoresce green when we shine blue light on it. This would make, for example, the mitochondria much easier to study because we can actually see them and what they are doing. So this is essentially how it would work. So we have the start codon with the first red arrow, so that would be AUG, and then everything after that would be the normal sequence for what HSP60 is supposed to be. Normally, HSP60, the gene for HSP60, ends with a stop codon. Well, what if instead of that stop codon, we just include the coding sequence for green fluorescent protein? The ribosome, as it starts at the start codon, it's going to start making HSP60 as it moves from left to right, 5' to 3'. It ordinarily would have reached a stop codon right here, but because we remove the stop codon and replace it with the sequence for GFP, it continues attaching amino acids to the growing HSP60, and now it's starting to make green fluorescent protein. And it will continue doing so until it reaches the natural stop codon at the end of GFP. So what we have produced here is the HSP60 protein that is attached to the green fluorescent protein. We call this a fusion protein. Basically, this is an extra large protein that just happens to fluoresce green, which is a really cool thing that we could do. So once we have this, we can insert this into a DNA vector and introduce it into cells, and they will start making this version of HSP60 that has GFP attached to it, and it should start making the mitochondria glow in the dark when we shine blue light on them. So you can actually see that here. These are mitochondria that have GFP attached to HSP60, and you can see that they are definitely green, as the name GFP promises, right? So this is just an example of the whole wide world that recombinant DNA technology opens up to us. This is not the only thing that you could do. The nice thing about biotechnology that makes it so exciting is as long as you have a good understanding of the foundational sciences like biology, chemistry, and so on, the only limit is your imagination. As long as you understand how the technique works, you can get cells to do just about anything, and that is what makes it so exciting. So that is going to be where we wrap up this chapter. I hope you enjoyed it. We are going to continue learning about these sorts of things as we move along through the course, so I hope you look forward to it. I know I do. I will see you next time with Chapter 3. Bye-bye.